Good morning. Thanks for having us. You will notice the name is not the same as in the program. My CEO couldn't make it today. He suggested I mention that we had the Boston Marathon yesterday and let you believe that he actually ran it. But the reality is the most running he did was to the airport and he didn't make it. So I will try to cover as best as I can. LogicBio is a genome editing company that is focusing on pediatric diseases. That's our positioning. And the reason we're going after this space is twofold. We believe it is large. There's a high unmet medical need. And at the same time, despite the breakthroughs and the successes that we've had in gene therapy and genome editing, pediatric patients offer a number of special situations that we believe are best addressed with our proprietary technology called GeneWrite. GeneWrite offers two big advantages. It offers a durable expression of the transgene. It's a stable integration, and because we are not episomal but actually go into the genome, the expression is not diluted as a rapidly dividing tissue is growing. And at the same time, it is a very safe approach because we do not rely on external promoters. We do not bring promoters with us in the AAV capsid. We do not rely on exogenous nucleases. We use a natural process called homologous recombination. And we believe that we have a very high site specificity of integration, and we can deliver all the hardware that we need for our treatment in a single capsid. So how do we do this? This is a technology that was developed out of Mark Kay's lab, who's the chair of human genetics in Stanford. And basically, our drug is an AAV-delivered DNA sequence that has the transgene. It has a 2A peptide, which is a 20 amino acid coding sequence for a self-folding peptide. And then it has flanking regions that are homologous to the albumin locus. These homology regions are between 400 and 1200 base pair long. And because of that extensive region of alignment, we believe that the site specificity and the risk of misalignment is very low. Through the process of homologous recombination, we can bring our construct into the genome and end up with a fused gene where we have our transgene just downstream from the albumin gene, separated from it by the 2A peptide. Upon transcription, you end up with a polycistronic messenger RNA that contains both the albumin gene and the transgene. And due to the process of ribosomal skipping, upon translation, you end up with two proteins. You end up with a slightly modified but still fully functional albumin and our transgene. So what makes this process so special is, again, you will notice we did not bring an external promoter. We rely on the strongest promoter in hepatocytes, which is the albumin promoter. As you probably all know, 50% of all protein content in plasma is albumin. There's very few promoters who are stronger than this. And we did not bring any exogenous nucleases, which, because they're mostly bacterially derived, can result in immune response. This technology was, as I mentioned, developed in Mark Kay's lab in Stanford, but we have since 4 additional preclinical proof of concepts, all of them done in different academic labs. It's a very powerful technology that works in the hands of many. And so far, we have proven that it can work in MMA, Crigler-Najjar, alpha-1 antitrypsin, neutralizing antibodies for HIV, and hemophilia B. We were funded in 2016 with the support of OrbiMed Israel. Our Series B was completed last summer with the help of OrbiMed US, Arix Biosciences, Pontifax, SBI, and again, because my CEO is French, we had to bring a little bit of Frenchness in and brought Edmond de Rothschild. We have declared our first development candidate. It's in the area of inborn errors of metabolism. We have yet to disclose the indication, but we have started CMC manufacturing. Our plan is to have an IND filed by the second half of next year, first in human by the end of next year. And because of the nature of what we do, and again, because as you may recall, we had the 2A peptide fused to the albumin protein, we have a natural biomarker to our process, so we can very quickly read out of patients what kind of efficiency we're achieving. So we should, by early 2020, know if the proof of concept that we have actually translates to humans. I think I'll leave it at that.
Great, thank you. Great, thank you. So I think a good place to start is maybe I could ask you the GeneRide technology itself. How would you compare and contrast that specifically with the other gene editing approaches, zinc fingers, talons, CRISPR, and where are the advantages with GeneRide?
So I think one of the big advantages, again, is that we don't require any external hardware. So if you have a zinc finger, you need at least 3 different delivery mechanisms or 3 different vectors to bring all of the components in, one for each of the zinc finger proteins and then your actual gene. When you're talking about CRISPR, you have to deal with the Cas9, you have to deal with a lot more hardware that we don't require. So with a single vector, we deliver everything that we can. That increases the efficiency, that increases the simplicity. And compared to the gene therapy approaches, we integrate. And again, if you look at any normal gene therapy, if you look for, there's an interesting experiment out of Jim Wilson's lab where he took a green fluorescent protein, put it into mice as gene therapy with a episomal expression. And you see that after 7 days, you have a very high expression. After 21 days, it's almost completely gone. And that just has to do with the dilution effect that you have in rapidly growing animals and dividing cells. As Kathy Hayes said this morning, in certain tissues in adults, that may not be an issue. There's experience of fairly constant expression over years. That's not the case in kids. And we work in our animal models, we work with neonate mice. These are mice that, when they're born and treated, weigh about 1.2 grams. And they grow to full-size mice, and we can maintain very stable expression. So those are, I think, some of the differences.
That's great. And you mentioned integration being a key aspect that differentiates it, and looking at pediatric populations. How does that then influence when you think about the efficiency of the integration initially and how that changes over time in a growing individual? Is there a selective advantage for the cells that have been integrated? How well does it persist? Does the efficiency change? Let's say you get X% integration initially, would that be the same percent integration or ability to detect it over time?
Yeah, so because our technology, as long as we're targeting the liver, it doesn't matter what transgene we're trying to bring in, we would still be going after the albumin locus. And going into the albumin locus, we can expect pretty much the same level of efficiency regardless of what the transgene is. We have a best-in-class capsid, which is LKO3, which is a capsid that actually Spark has brought into the clinic, and we have some clinical safety data there. But for any disease that would target the liver, we would use the same capsid, the same homology arms, get into the same locus, and expect a similar level of efficiency. That level for us at this time is about 1%. But I think what's important to understand is that it's 1% into albumin. And albumin, again, being such a strong promoter, so highly transcribed, with a 1% transformation of the albumin locus, we get to about 15 to 20% physiological levels of expression for factor IX. So it's a big difference whether you bring in your transgene into the original site of the defective gene, and therefore have to rely on the original promoter, which may more often than not be much weaker than albumin, but also have different levels of efficiency because epigenetically speaking, every gene will be less available for homologous recombination. We are going to be very consistent with that level of integration into albumin. We then have another aspect, which is in all of those indications where the disease places the cell at a disadvantage over healthy cells. Let's say you have an inborn error of metabolism where the faulty gene produces a byproduct which is toxic. The untreated cells or the untransformed cells will be outcompete by the cells that have received the transgene. And we have data in two models. One is alpha-1 antitrypsin, the other one is MMA, where we started with an integration efficiency of 1%, and over a period of 6 to 12 months, we increased that to about 15 to 20%. So you can either choose an indication where you will have a competitive advantage. You can actually induce or force a competitive environment, giving yourselves a competitive advantage. But even without that, we have 5 separate models where the 1% indication resulted in therapeutic effects, and we believe that that will be translating into humans. The expression level is very constant. We have seen mice stay at a level of the same level of expression of over 15 months, and that for a mice is about middle age.
Okay, and obviously targeting the liver, targeting albumin is the focus now. Should we think about that being the focus near term to midterm as well, or are you looking to expand the scope more rapidly than that to other organs? Does it ever make sense to target something other than albumin in the liver? Maybe you could comment on that.
So I think in the liver we're very well served with albumin. We are, however, not bound to that locus. We can select any locus of our choice, and obviously albumin would not be the one if we went into muscle or CNS. There are other loci that we have experience with where we have shown that we can integrate. The capsid that we currently use is also not one that has a high tropism for muscle or CNS, so we would also have to change that. And yes, we're very much actively looking for expanding the potential of GeneRight into other tissues. We have collaborations with academic centers to help us find those capsids, and we're actively working on selecting what loci we should use. But to be honest, at least initially, I expect the first 2, 3, 4 indications to be in the liver just because that's where we are currently now best positioned to work.
And have you thought about the potential of the technology, obviously, to deliver a transgene? You could imagine using this as well to introduce shRNA or just to disrupt gene disruption. I don't know if you can use this for gene correction as well. Have you thought about those different applications and approaching those with the technology?
Yes. So first, gene correction might be a little bit more difficult because, again, we're targeting the albumin locus, so we're not really changing the original defective gene. But there are indications, and we actually have experiments that have been published, where on top of the transgene, we introduced shRNA or any kind of interference RNA to not knock out, but knock down the defective gene product, especially in those cases where, while the corrected gene will confer an advantage, the persistence of the gene product of the defective gene confers a disadvantage. So you could introduce both the homology arms of the transgene going into the albumin locus, and then a way to knock down the defective gene. Again, gene correction I don't think is high on our list at this point.
Okay, and on the slide, it's showing here, obviously, the IP that you've licensed and expanded in 2018. Maybe you just wanted to comment on what your IP is covering and your capacity there, your capabilities.
Yeah, so we have a full set of IP coming out of Stanford. There's 4 patent families covering promoterless and nuclease-free genome editing. It is not bound to any locus. We have experience and data for several different loci. It is actually also not bound to any specific capsid. But we do have also, separately from GeneWright, a portfolio of IP on capsids that were also developed in Marquet's lab that resulted from capsid shuffling, generating a number of synthetic capsids and then screening them using mice with humanized livers. That's how LK03 was actually developed. And in this case, while we have exclusive rights to GeneWright, we have exclusive rights to these portfolio of capsids for GeneWright, non-exclusive rights for general gene therapy, and that's how Spark actually got their hands on LK03 and took it into the clinic. And we view that as actually as a very positive aspect, because now we're going into the clinic with a capsid that has already been proven.
Great. Matthias, thank you.
Thank you.